Hello and welcome to another screencast where this time we'll be discussing cardiovascular regulation. And I think we're gonna break this into three different ones here. So in this first one here, we'll be describing and explaining the need for regulation of perfusion, and then briefly just talking here about the mechanisms involved with the regulation, and then the autoregulation mechanism here in this screencast. And then we'll deal with the neural mechanisms and the endocrine or hormonal mechanisms in another one. So the tissue or cell activity in the body changes, so perfusion is going to need to change. That is, that perfusion will then need to be regulated, right? Because you have, you see a capillary bed here. Here's your small artery, right, and the little arterioles, and then these are all the little capillary beds here and here. And so what you're not seeing is all the cells, but the cell's demand for oxygen and the production of CO2 is going to change depending on the activity of those cells. So blood flow to these areas will be either stimulated, right, or will be cut off if you don't need that blood to be delivered there. So tissue perfusion is important, right? And of course the factors that are affecting that are the cardiac output. That's not right in there. Cardiac, so we'll just do it over here. Cardiac output, your peripheral resistance, and then blood pressure. Right? So these factors are kind of being monitored there by the nervous system, by the endocrine system, and making adjustments there. So why do you need to regulate this again? So that you get blood to that area at the right time. Not only at the right time, but you want to get that blood at the right area, right? So it's at the right time, right? At the right area. And then you want to move that blood without drastically changing or affecting blood pressure, and then perfusion to vital organs, brain, heart. So you can imagine if you all of a sudden open up all these capillary beds in the skin, then you would have a great rush of blood to these areas, and then peripheral resistance goes way down. You then have to move that blood away from other organs, vital organs, etc. Blood pressure drops, etc. And then you don't get oxygenated blood to the brain. That's going to be a problem in a few minutes, right? Or a few seconds, rather, not even minutes. So the first mechanism that is involved with doing this is this auto-regulation method. And this auto-regulation is local, right? Meaning at the capillary beds themselves, at capillary beds. So these mechanisms are in place locally to change the pattern of blood flow. So it would be changing this pattern of blood flow, opening up, again, as I said, some of these capillaries and then closing off other ones locally here. So the stimuli to do this, right? So this is so let me just back up a second. This is the bigger picture, right? So we're going to have these auto-regulation mechanisms that we're going to discuss here, and then you have neural and hormonal mechanisms that are also in place. But we're going to, again, this one here, we're just dealing with this auto-regulation. So if we look at this a little bit larger here, this homeostasis is in some way disturbed, right? So we have some sort of physical stress or trauma, chemical changes, right? Or increased tissue activity, right? Increased or altered, changed tissue activity. In this case, they're talking about having an increase in this tissue activity, right? So this follows and makes sense, right? So if you have increased tissue activity, you're going to have decreased O2 because the tissues are using those O2, right? Increased tissue activity. Lowers O2 because it's being used. This will also increase CO2 because as the cells are using that oxygen, performing cell respiration mechanisms, you're going to be producing CO2 as well. Right? and then this pH is also going to go down, which we'll address in another screencast there. So these are cues, these are stimuli, right, that will then sort of signal that there's inadequate local blood pressure and or blood flow. Right? So this is corrected by this local decrease in resistance and increase in blood flow. Again, this local decrease in resistance would be due to vasodilation. So if we get vasodilation happening, the capillaries go from either, well, this, right, completely closed off, right, to this, to this. And remember that as we increase that diameter, the friction goes down, and then we can increase the flow there, lower peripheral resistance, right? So if we do that, that is then going to bring more blood to those capillary beds. Right? More blood means more oxygen and more CO2 carried away, so your CO2 levels are going to go down and your pH levels are going to come back up then. So then homeostasis restored back to that normal blood pressure or volume. So let's just take another look at this. A way to sort of, when I'm describing this, this is what I'm thinking about, is that I have these section of a precapillary sphincter right here, and this sphincter is a circular muscle group here. And so when these smooth muscles contract, they will actually make the diameter of this blood vessel go down. When they relax, you get the diameter increasing, right? So these smooth muscle cells are the ones responsible for vasoconstriction and vasodilation. Okay. So when we, again, the increased in cellular activity, so if we have cells that are over here that are increasing their activity, right, that is going to lead to vasodilation to get more blood delivered to those cells to bring more oxygen, right, and then any CO2 that was being dumped off, that CO2 will then be able to leave that area right quickly. Right. Vasoconstriction then would be the opposite, right? If there's too much blood going to these cells, to these cell areas here, then the O2 levels are pretty high. That is, and CO2 levels would be low. These cells here not active, or not as active, I should say. Not as active. The O2 levels would be up there, CO2 is down, so you don't need to bring the blood this way, so you would close off these are the smooth muscles here, they would close off or vasoconstrict, and then divert blood to the capillary parts that are needed there. So the main stimulus for vasoconstriction, right, is increased oxygen levels. The main stimulus here for vasodilation, right, would be lower oxygen levels, higher CO2, and lower pH levels there. What also might trigger this is if you had any sort of damage to the blood vessels. So this damage will, the cells that are damaged there will send off NO, which is nitric oxide. And damaged cells will also release histamine. And nitric oxide and histamine are two chemicals that will also signal these smooth muscles there to relax and then vasodilation. So if you have damage to the area, you would want repair factors, you would want white blood cells to come to this area to prevent any sort of infection, so you'd have increased flow to that area of damage, right? So these are also factors that can affect that. Now, all of this autoregulation and all this blood flow changes here may make it seem like you're greatly diverting lots and lots of volume from the rest of the system, and you would decrease blood pressure or whatever. And it's not really true. Most of this is probably not even really detected. You probably don't even have an extreme variation of peripheral resistance here, because again, this is all local. These are relatively small volumes of blood that are moving, right? But and this is kind of going on all the time depending on the activity of those cells in different areas, okay? So again, these autoregulation mechanisms involve monitoring basically oxygen levels, carbon dioxide levels, and then any other sort of chemicals that might be released because of damage, right? So hopefully you should be able to now kind of pause and briefly describe and explain the need for this regulation of perfusion. Identify the three mechanisms that we're going to discuss here, and the first of those, again, was the autoregulation mechanism, and be able to describe and explain a little bit about what's going on there. Okay, thanks for listening. Bring your questions to class.